So hi everyone. Today I'm going to talk to you a little bit about our recent work. The goal is to disentangle cell type associations, and we have this. The methodology can actually apply to a wide range of diseases, but I'm going to talk a little bit about two case studies in neurodegeneration. So the kind of overarching kind of question that we're really interested in here is that there are lots of kind of the growth of large genomics data sets or kind of omics data sets at large. But how can we creatively integrate them in new ways to kind of derive potentially more insights than either of the pieces themselves? So one of the data types that I'll be talking a little bit about today, some of you may have heard of, it's called genome-wide association studies, or GWAS. And this was actually kind of a category of studies that have really kind of came out of after the Human Genome Project, where the basic idea is that a lot of us have actually a few areas in our genome that distinguish us from one another. The majority of the genome is actually very similar. So as we were able to sequence or probe these differences in the genomes over time, there's a natural question. If we have people with, a lot of people that eventually have a particular disease, Alzheimer's, cancer, many, whichever disease, you can collect a large group of them, call them cases, and then, also kind of enroll a lot of people that are, what seem normal and healthy and do not have the disease of interest. And then the idea is actually simple, right? You compare the genomic variants between these two groups and look for which variants seem to be over-enriched in the cases, and maybe that's a signal that these variants might be associated somehow with disease risk eventually. And so this has been actually very powerful. It's led to a lot of interesting discoveries about, which parts of our genome might have, coding for additional risk towards particular diseases. So I give you some examples. There are lots of GWAS studies. So Alzheimer's disease, autoimmune diseases, schizophrenia. But I wanted to point out also that it's not just for diseases. People also do these types of studies for kind of complex traits. LDL level, whatever in your blood panel that you might often measure. People even do it for eventual college education attainment, right? So just to see, are there potential links between, variants in our genome? And of course, the people who run these studies try to correct for all kinds of, covariates or other batch effects that might be kind of linked. One of the limitations, though, of this data type, they're just looking at genomic variants in different individuals. So inherently, there's no sense of tissue and cell specificity. So I think in the intro, we already mentioned that I'm very interested in this question because ultimately, if you think about it a little bit more carefully, in every cell, most in your body, you start with the same DNA, really, right? And then, of course, everything becomes kind of different and there's different function. And the thing is that with disease, we know that different cell types can have different interactions with disease. Most complex diseases do have some tissue and cell specificity. So having a better understanding of which tissues and cell types are linked with disease is actually a super fundamental question. And surprisingly, for a lot of these complex diseases, we don't even have a really good understanding of which cell types are the ones that are implicated. And that's like a big area of research in a lot of diseases. And so that's why I'll pivot to kind of thinking a little bit about neurodegeneration, because this is an area where the complexity of the brain becomes this kind of huge additional adds a lot of additional complexity in studying tissue and cell specificity. So, we already know that, our brains are really, really kind of complex. They have lots of different regions. The different regions kind of dictate different types of functions. Beyond that, if you think about regions, we know there are tons of different types of neurons, and these neurons can have different interplay, but the circuitry matters a lot in these different regions. Beyond neurons, there are also accessory cells called glial cells that play very important roles in the brain. So this is, we're not going to talk about, necessarily go into the details of brain anatomy or kind of the cell types implicated in we will talk about the cell types implicated in different parts of the brain, but the whole idea is just this complexity is still being probed and understood. We don't have a good grasp on kind of these kind of fundamental differences in the brain. So what do we have, though, right? I just said that, everything is super complicated. We have lots of brain regions. We have lots of cell types. We don't really fully know what's going on. The rise of single cell technologies has allowed us to now probe and get very high resolution data of the brain at unprecedented resolution. So this means that basically what people are doing is collecting different brain slices, disassociating them and measuring the gene expression in individual cell types. Note that, there's a lot of complexity in this process as well that I'm going to kind of just, gloss over. But the idea is that we can get measurements at the single cell level. People then try to go and label these cells, et cetera, et cetera. But we actually now have a better sense of what the gene expression or changes and variations are within kind of the brain. So our big kind of question is, we have these disease variants, this GWAS data type, not tissue or cell type specific, but captures very valuable information about disease. And now we have, all of these brain kind of cell type, region kind of expression levels. Most of these are being collected from normal, post-mortem brains. Is there a way we could kind of intelligently kind of integrate them together to reason a little bit about the cell type and tissue specificity of, in one of the examples downstream, neurodegenerative diseases, but really any disease kind of that we have a good collection of single cell data. And so I wanted to point out that this is, of course, unpaired, right? We're taking population level studies from a lot of people and then single cell data from a select few of individuals. So we come up with this problem is not, we're not the first ones to think about this. I'm just showing a kind of general workflow that a lot of these methods that are thinking about this question of identifying cell type and trait association, so that's the endpoint kind of down here, what they kind of generally look like. So I think, so basically I discussed what GWAS is, this variant trait association. One common way to process this is to collapse these variants into gene level scores. There's a very popular method called MAGMA for this. The details don't super matter in this case. And then we have the single cell RNA-seq that's kind of the cell type characterization, right? So all of the methods are trying to kind of tackle this question of how can we integrate these gene trait association scores with cell type expression. So when we kind of dug into this at first and looked into all of these methods, we found that there were several kind of practical and limitations essentially towards kind of systematically and robustly deriving these cell type trait associations. So I'll just mention some of them. Typically, there are just methodologically a lot of kind of thresholding that people have to choose, which means that practically people just try like a very many different thresholds and then try to, figure out which results they think look the best. So there's tons of thresholding. And then in terms of we want basically a kind of hypothesis testing at the end so we can get a p-value for these cell type trait associations. There are methods that kind of have to do this through kind of very involved simulations. And so we'll see this, that what this means is that it becomes very computationally expensive and difficult to run when you have a lot of data. So I wanted to kind of just discuss our framework kind of briefly before I get into some examples. So one of the kind of big insights of our method also is that especially when looking at the brain, so you remember I told you that we have like brain regions, we have brain cell types, we have different cell types. You can actually classify kind of or group cells at different levels of granularity. So this is something that hasn't been kind of explored as much in current literature. But we actually think that this is actually a very important distinction because you can imagine that there are some kind of traits that are more kind of associated at the region level, some more at the cell type level, or some at the interaction or intersection of these two, right? So specific cell types in particular brain regions. So what we really do, and I'll get into kind of our specificity score later, is that we develop kind of a novel specificity score that actually captures the kind of cellular identity at a particular kind of resolution. And then we have a regression-based association where we can basically get a score for every cell type and trait, assess the statistical significance. And another aspect that no other previous method has looked at is what we've decided to name influential gene identification. So what is this? So a lot of people are interested in this question of cell type trait association, right? But for us, we are very interested in what the underlying molecular mechanisms that drive disease. So once we get the cell type and trait, that's very interesting, but it still doesn't fully get to the question of what are the molecular mechanisms that drive that association? So what we actually do is come up with a clever way to identify the specific genes that drive a particular cell type and trait association, because once we have genes, we can assess which pathways and get a little bit more closer to generating a hypothesis for what the association might be. So I'll just so I won't walk through any of our equations. I just wanted to kind of highlight the specificity score is really the driving force behind our seismic framework. And the intuition is actually quite simple. What we realized is the characteristic of a cell type is actually, at the gene level, is both whether the gene is higher expressed and whether the gene is present. So I think this is actually an interesting distinction to think about, right? Because oftentimes when people do differential gene expression, we're really looking at differential abundance. And so we thought that, to really characterize a cell type, we actually have both the binary signal, presence, absence, and also the actual expression abundance. So this is basically like a clever way to combine them into a specificity score. Again, I'm happy to kind of talk about this more if there are any questions. But I just wanted to kind of jump to kind of the results, right? So what happens after we kind of develop this method? Of course, the first thing we wanted to do is this is actually often used to, get p-values and statistical kind of associations afterwards, right? We want to make sure that the p-values we're estimating are not overinflated, so to speak. So we do a ton of null simulations, which basically means we make sure there's no disease signal in a simulation study and check that our p-values are kind of as you expect, right? Based on the definition of a p-value. We're not like inflating them. And then, of course, we also can do simulation studies using a sophisticated single cell kind of synthetic data generation mechanism to explore whether we have very good power. So what I wanted to highlight for these results, these are kind of, we compare against 3 existing methods that have several of the limitations I mentioned before. This SEDRS method is the kind of most recent kind of state-of-the-art method. And we show that basically we have comparable, if not a little better, kind of type 1 error assessment, which is our p-values are kind of conservative or not inflated. But I think what's interesting about the power analysis that we found is that if you look kind of closely at this, we are very comparable to SEDRS when we simulate a large expression fold change in the disease genes. Where our seismic method is really strong, which is actually what we think is more representative of a lot of complex diseases, is when the disease genes have actually just a very, very small expression fold change. So of course this is much challenging, right? We're talking about like say sometimes a 0.05 expression fold change. Can we still pick this up and identify the relevant disease? And so of course power drops for everybody, but what we see here is that we can actually still kind of fish out some signal even when that expression kind of changes is quite low. So, and then one other kind of point, as I kind of mentioned earlier, so SEDRS is a method that uses lots of simulations to basically kind of assess its statistical significance. So the other kind of big limitation here is that it's much, much slower than all of the other methods that we're looking at, which really kind of hampers its deployment kind of to systematic study. And this is kind of especially kind of as the number of cell types increases, which is the natural kind of trend of things. As with all data these days, there's more and more data being collected. So, beyond these simulation studies, of course, what we're really interested in is looking at real data, right? And I mentioned that, while we're really interested in this question of studying neurodegeneration, we can actually apply this method to any GWAS data and any single cell transcriptomics data. So what we do kind of first is collect an assortment of kind of different GWAS studies, in this case 27 that fall into neurological or immune diseases, and as well as kind of complex traits, so to speak, and including kind of smoking, BMI, college education, et cetera. And then we take a large kind of collection of kind of atlas scale single cell transcriptomics data. In this case, the single cell transcriptomics data is actually collected from mouse, and so our method is actually able to take advantage of single cell data from mouse. Of course, we can do it easily for human as well. The reason why we often kind of choose to use mouse is actually because the mouse data is usually going to be higher quality than human, but we also have a similar results for the human level data. And kind of what I wanted to just kind of, this is, we ran kind of each of these GWAS studies against our kind of trait cell type association method, seismic. And what the heat map is showing is essentially the brighter the color, the more significance we're assigning to it. And basically, I think there are some kind of interesting results kind of already if we look at this, right? So, some of the things that are kind of more intuitive, because of course, like the neurological disorders, we're finding more associations in brain, but if you kind of look closely, we actually find kind of, of course, several of them have neuronal associations, but this is not consistently across the case for every disorder. And then, of course, the immune disorders, we find a lot of blood immune kind of related associations, but the blood count related traits, we also find associations in the blood immune kind of area. And then, of course, what we thought was interesting is this association with the pancreas with several neurological disorders, which actually matches some of the findings that people are starting to get from recent kind of studies and the links between diabetes, for example, with neurological disorders. And so, it was actually fun to kind of look at this heat map, dig into it a little bit and be like, okay, are there, what are kind of new findings here? Or, a lot of things are actually already recapitulated by literature. And so, we also, of course, wanted to compare with the existing methods that I had mentioned. And, I still want to say that, like, a lot of them are actually quite correlated with the findings in kind of previous studies. I think our method is just kind of bringing kind of additional power and interpretability over some of the previous methods. But we actually pick up a lot of the findings that they do also. But so I wanted to jump in, of course, to the goal that I mentioned at the beginning of the study. So looking at, associations with complex neurological or neurodegenerative diseases. So this is actually where I think arguably we kind of sometimes know kind of the least about kind of associated cell types. And this is still like a very active area of research. So we first tried our method comparing a large brain kind of a collection of brain cell types against Parkinson's disease. And basically, we also compare with other methods, and we find that several of the methods identify associations with dopaminergic neurons. And if you're kind of familiar with a little bit of kind of the mechanisms underlying Parkinson's, this is known that dopamine levels and dopaminergic neurons have some kind of association with Parkinson's disease. But, beyond that though, remember I was saying that we are interested in thinking about, different resolutions or different granularities that we can assess. So we already know that dopamine is associated with Parkinson's. What we really want to do is dive deeper, because we know that there are dopaminergic neurons in several regions of the brain. So can we actually add in that brain region specificity and further assess kind of what's going on and what are these associations? So that's actually what we do. We actually kind of can split the same data up into 5 different levels of granularity. So what I showed you earlier was actually just kind of subclass level, which is kind of the class of neuron, right? Dopaminergic neurons is a class of neurons. They're actually spread out through the brain. So, the first thing we can do is actually combine kind of brain region and class, and then, you get higher and higher levels of resolution. So at the subclass level, we actually are only looking at 14 different cell type characterizations, and you can kind of go down kind of more and more and more specific until you can actually, from this, eke out 231 different cell type characterizations in the same data set. And so we call this brain region fine cluster, because that's actually what the single cell folks do. We don't actually label the cell types ourselves. We take what the kind of experts who collected the data have annotated the cell types as. And so kind of what I'm hoping is kind of what you'll see is that the results are consistent, specifically for our method, seismic, that top method, across granularities. But when we kind of go down in specificity, what we're actually doing is finding more and more high resolution results. So we actually not only understand that it's dopaminergic neurons that are finding this association, we can then see that there's this kind of specific subtypes of the dopaminergic neurons in the substantia nigra, which is a region that we know degenerates in Parkinson's disease that have a strong association. So, so this I think was really exciting, and then so we wanted to actually move also and think about, can we use this method for Alzheimer's disease, where if you've actually been following Alzheimer's disease, GWAS associations or the research in this area, there's actually been something that's kind of an interesting kind of debate, so to speak. So what people have found through Alzheimer's disease GWAS studies is that a lot of the genes in these large clinical GWAS are all implicating no neurons, only kind of the brain immune cell types, microglia. And we see that, if we use these large clinical GWAS, we do also see these very, very strong associations with these microglia. And so there's been actually a lot of debate where people are like, is actually Alzheimer's just an immune disease? Like, should it be even be considered like kind of a neurological disruption anymore? Is it just, so there's actually a huge debate in that respect. And so actually collaborating, we have a kind of a collaborator who specifically studies Alzheimer's disease, who had the critical insight that, maybe, everybody's using these giant clinical Alzheimer's disease GWAS studies, but we actually don't have good kind of diagnosis, like kind of clinical diagnosis capabilities, in the sense that there are likely several different subtypes of Alzheimer's disease, and Alzheimer's disease has huge interaction effects with environment that we actually maybe, should think about using different endpoints. And so, so this actually comes from the kind of inspiration that also we know that in Alzheimer's disease, different brain regions do seem to still be associated with Alzheimer's disease. So, many of you probably know in Alzheimer's disease, we see kind of memory dysfunction very kind of as one of the first symptoms. And that's actually very much related to the brain region that's impacted and degenerating in Alzheimer's disease, so the hippocampus or entorhinal cortex. So when we actually choose a specific pathological biomarker-based endpoint GWAS, even though there are many, many orders of magnitude fewer individuals kind of analyzed in this GWAS, we do actually see exactly the neurons that are vulnerable in Alzheimer's disease being implicated by our method. And so this was extremely exciting for us to see, because people have never previously been able to associate an Alzheimer's disease-related GWAS with a neuronal signature from the exact biological neurons that we know are related. But you might say, oh, hippocampus, we already knew hippocampus is degenerating, right? Like, so what are we gaining from this? And this goes back to what I mentioned early on when we were looking at the workflow. What we're really excited about is this idea that we can then take the influential genes driving the association and look at what they are. And so this is actually what we're doing now. We can find that entorhinal cortex association or hippocampus association, look at the genes that are driving those results, and hopefully better understand why these cell types are particularly vulnerable to Alzheimer's disease. And so this is ongoing work that we're doing with our collaborator. And so I just wanted to kind of wrap up and acknowledge, of course, my lab, and then our Alzheimer's disease collaborator who is at Boston University.
Thank you, Dr. Yao. Any questions?
I have a question that it's usually it's the macrophagia that people encounter that is working. Why there is no more studies of pathogens or virus or something studying what they are there? Because usually it's an immune system trying to attack something.
Yeah, so I think that's actually a great question. I think actually if you've, in Alzheimer's disease, people do have really wanted to see if there is actually a pathogen-related reason. So people have, found these noisy studies that it seems like there might be some association with herpes simplex virus, for example, but there's no actually really, I think it's just very, very hard to study if we think about, Alzheimer's disease is very age-related, and, maybe it could be some latent infection kind of that are kind of sustained through time. So I think there's just not actually so much there are a lot of hypotheses. People are trying to work on it, but there's not, I think, a very clear support for like a particular, viral or pathogen infection, I think, generally yet. I do want to say that I think the microglia, yes, it's an immune kind of cell, but it also actually does so many functions in the brain that it's not quite fully the same analogy as just like only, it does so many more functions in terms of maintaining or helping the health of a neuron than just kind of immune related functions. So I think it's also kind of an additional added layer of complexity.
Hi, it's a very nice talk. So I have a question, like you mentioned that you associate with mice data as well as UVAS data.
Yeah.
Like how did you actually, from UVAS, it's like human source.
Yeah.
And from mouse specifically, it's completely, they have different, there are significantly different difference between these two species. So how did you actually make the significance association between these two data sources?
Yeah, so right now, in terms of how we use the mouse kind of transcriptomic atlases, we're doing kind of the very simple naive thing of kind of mapping mouse genes to their orthologs and then to their human orthologs and then doing this comparison. Like I mentioned, we are also very excited. We actually also have applied the same method to the human kind of single cell atlases. It's just that the data quality is actually much lower. So of course, I think the goal is eventually to kind of, especially as the human kind of data gets higher and higher quality, to kind of leverage that more. But especially in kind of brain, I think there's just way more mouse data kind of currently available, I think just kind of as a function of data collection efforts. Yeah.
Dr. Yao, thank you so much for this wonderful talk. This is really, really enjoyable on a number of levels. I have a question. You mentioned that seismic runs a little bit slower relative to one of the other algorithms, and I was kind of curious as to, was that because sort of decomposing I pulled up one of the papers that you had up here, and I was kind of looking at the nice elegant algorithm that you have for that. What do you think is slowing it down? Like, is it the variance computations there? Is there has there been any interest in where it's slowing things down, such that there could be potential modifications in the future to speed it up and still get similar performance?
Yeah. I think maybe I misspoke when I was on the slide, but I think I was saying SEDRS, which is the current state-of-the-art method, is the one that's really slow. Our seismic method is actually very fast and comparable to like some of the early kind of simpler methods that were also very fast. So
I'm sorry.
Oh, no worries. So in this graph, our method is in red, so seismic. I think, maybe once we get to a lot of cell types, we're like a smidge slower than some of the existing methods, but I would say it's very comparable performance.
I apologize then.
Oh yeah, no
I
worries.
misunderstood it.
I think, and I can say I know why this SEDRS method is slow is actually what they do is they do the cell grouping at the last stage. So what they do is they find individual associations with each cell, not each cell type, but each cell in the atlas data, which you can imagine that's a very slow process. And then they only kind of collapse that analysis at the end when you give it different groupings.
Other questions? Great questions. Thank you, Dr. Yao.
Okay, thank you.